Great. Thanks so much, Hannah. And I'm so excited to be with you this morning and to review some of Mission Bio's publications. We actually decided to break it up into two parts because we want to really be able to highlight the aspects of all of our publications and give them justice. So this will be part one, and we'll hold part two later in June. Mission Bio has been around since 2017, and we often get asked, how is Mission Bio different from other single-cell companies? And the main point is that Mission Bio is a single-cell DNA sequencing company. We were the industry first single-cell DNA platform, which we launched back in 2017. And we launched with a couple catalog panels in AML and solid tumor to really get our customers going. Well, in 2018, we launched Tapestri Designer, which unlocked the potential to make custom targeted DNA panels on our platform. And that really opened up the field to not just heme and solid tumor applications, but really to a wealth of any applications that involve DNA. Including CRISPR screening, mouse models, and so forth. And then more recently, last year in 2019, we've been hard at work to launch the industry first DNA multi-omics platform, in which we combine DNA and protein from the same single cell. And so really this launches us into the preclinical and biomarker space, and we're really excited about adding this new capability to the Tapestri platform to drive it from a DNA platform to a DNA multi-omics platform. So today I wanted to go over a couple of our seminal papers in the field. The first publications really started profiling, looking at AML samples and starting to look at them using our single-cell DNAseq technology to understand more in more depth, what is the clonal architecture underneath these samples. So we'll be going over Mission Bio's first couple publications. We'll take a sneak peek into DNA plus protein. And then, as I alluded to earlier, in part two, which will happen next June, we'll really dive into the pharma and clinical aspects that the Tapestri platform has been used for in some key clinical studies, looking at AML treatments or solid tumor treatments, and delving into whether the Tapestri platform is good for MRD detection. All right, but for today, I'd like to start talking about how the Tapestri platform has been used in AML, and really to retrospectively go back into that freezer and take AML samples and start to interrogate them on a single cell level to figure out what more information can single cell genomics give you. Just a little background on AML, for those of you who don't know. AML is a blood cancer, and it arises from immature myoblasts. So typically your red, white, and platelet cells are made in the squishy tissue of some bones called the bone marrow. And it's called AML because it's acute or fast growing. And once the cancer begins, it moves quickly from the bone marrow into the bloodstream. Now, AML affects the myeloid cells that differentiate into the red, the white, blood cells, and the platelets. However, AML is particularly affects the white blood cells. And so what happens in AML is that these myeloid stem cells, instead of differentiating all the way into a mature white blood cell, they become stuck in an immature state called a myeloblast. And these are abnormal, and they build up in the bone marrow and they build up in the blood. And so when you actually go and diagnose AML, you look at the percentage of myeloblasts in the blood in order to have a diagnosis. So AML is a blood cancer of white blood cells. And how can single cell technology be applied to take a closer look at cancer. Very recently, Nick Navin's group from MD Anderson just did a really beautiful review in Cancer Cell. And so here they do a really nice overview of single cell genomics technology. They look at DNAseq, RNA-seq, and ATAC-seq, and they show where it can be applied in cancer research. So on the top left, you can look for cells that, very rare cells that have cancer mutations in which the cancers arise from. You can take these mutations from the cells or RNA expression signature profiles and create phylogenetic trees in order to look at how the clonality evolves with time or with treatment. You can look at the tumor microenvironment of where the cells are growing in the normal tissue. And this is where we're really excited about pairing our DNA plus protein capabilities, because the DNA will be able to identify the cancerous cells, while that protein information will really give you the information about the cell type or identity within that tumor microenvironment. So a lot of powerful things by pairing DNA plus protein. You can see how the malignant cells traverse throughout the body and look at the different clonal architecture in different parts of the body at different tumor sites. And then we think this is going to be a really big application to the field, which is looking at how the mutations within the cells progress as patients are given different therapies and they go from clinical remission to relapse, they go from responding to not responding, really understanding the underlying molecular mechanisms that determine who goes on to respond to treatment and who doesn't. And so on the right, in the big box, is just kind of highlighting what single cell DNA sequencing allows you to see. And the number one thing is the clonal architecture. So instead of having from bulk sequencing, knowing that I have KRAS and EGFR cells, single cell sequencing allows you to tell whether those are coming from the same cells or from different clones. And once you have the identity of the clones, you can create beautiful phylogenetic trees, understand the origin of how these mutations are acquired. Again, you can look at how they respond with therapeutic pressure shown on the bottom left. And then you can, you know for sure whether these mutations are co-occurring in the same cell, as well as the zygosity state of these mutations. So I really encourage you to check out Nick Navin's cancer cell review that just came out. All right, so to start, our very first paper came out in 2018. And it was the first paper that showed the power of single cell DNA sequence in a high throughput fashion. And the reason why we're able to do DNA sequencing at a high throughput fashion is because we do them in microlipid droplets. And I'm sure you've heard about the miniaturization of PCR or NGS reaction, really putting things in droplets as opposed to having a big well to do the reaction allows you to scale up immensely from 100 reactions to thousands of reactions. And so in this seminal first paper, they used the Mission Bio Tapestri Platform to look at 2 patient samples of AML. And they wanted to ask the very first question, what information does single cell DNA sequencing uncover that is lost with bulk sequencing? And this was done in conjunction with Dr. Takahashi's group at MD Anderson. So this first patient was a 65-year-old male with AML who had been given chemotherapy and he achieved complete remission. And so we had 2 samples. We had a sample at diagnosis and we had a sample at relapse, which is shown on the left. And so you'll, in this plot, each row represents a single cell and each column represents a different variant, cancer mutation. And so what you can notice is that first off, you can see zygosity at the single cell level shown in gray, maroon, and dark red. The other thing that you get is you can see co-occurring mutations. So whether a single cell has is wild type throughout all 3 alleles, or is het or home at 1 or 2 of the alleles. And so we were able to profile both at diagnosis and relapse thousands of single cells. Then you can group them into clones based on their mutations and co-occurring mutations, which is shown in the middle. And we show that this blue clone is the one that expands during relapse. And then on the right, we show that the data match up very well to bulk sequencing. However, with single cell sequencing, it goes down to 0.1%. And oftentimes you can detect clones or mutations at a higher sensitivity than you can with bulk, which is usually around the 1 to 5% range. In the second patient, they had the opportunity to look at 3 samples. So not only did they have the samples at diagnosis and relapse, but they also had a sample at remission. And so what's really very cool and interesting to see is that, again, we see this nice zygosity and co-occurrence of mutations. And during remission, that almost all the cancer cells almost completely go away and everything is almost wild type. However, when you look on the right at the clonal architecture, it's hard to see, but if you zoom in, there are 3 and 7 cells that are detected here at remission. And these cells go on to expand during relapse. And so this is really one of the interesting, enticing things about single cell technology is being able to use this for minimal residual disease detection. If you could follow patients at remission and actually detect what clones might still be there that have cancer mutations, you could potentially put them on targeted therapies much earlier on because you're able to detect these clones at very low levels. So this was Mission Bio's first paper in collaboration with MD Anderson to really just show the different uses of what single cell DNAseq technology can give you. In a second paper that came out very early on with our technology was this really cool idea that you can use the DNA sequencing not only to look for cancer mutations within cells, but you can actually look for normal mutations and use the mutations to look at genotyping. And so in this paper by Dr. Zender from Stanford Medicine, he looked at patients that had AML but were given bone marrow transplants. And so he had multiple time points. He had before the bone marrow transplant, after the bone marrow transplant, and then once AML had relapsed. And he asked the question, can you look at the cellular mosaicism between the donor and recipient to predict relapse? And then what are the benefits of single cell versus bulk? And so here we're looking at 2 patients. Patient 1 is on the top and patient 2 is on the bottom. And you can see pre-bone marrow, all of the cells are from the patient, which is shown in the black box. After bone marrow transplantation, you can figure out what the ratio is between patient cells and cells coming from the bone marrow transplant, shown in gray. And so at the top, there's about a 50-50 split between the patient's own cells and the transferred cells, versus at the bottom, that patient got more of the transferred cells in the bone marrow transfer. And then you can do this analysis again at relapse, unfortunately, and again measure this proportion between the patient's own cell versus the donor cells. And what they uncovered in this study is that patient 2 ended up faring better and relapse occurred much later than for patient 1. And so what they conclude is that the more successful the bone marrow transplantation was, so the more cells that come from the bone marrow transplantation, the better chances to have a longer time before relapse, which I think is a really cool, interesting way to use the Tapestri platform. And then when they looked further into patient number 2, they were able to reconstruct the clonality. They're actually able to show which clones come from the patient versus which clones come from the donor. And then in this instance, they are able to show that they think the clone in green, which is a double mutant for TP53 and SF3B, is the one that is potentially causing relapse because it's there at very low levels and it expands during relapse. So one, with single cell technology, they were able to detect this clone at 0.2% during this point when they thought the patient was fine. And then furthermore, with single cell technology, they're able to detect that both of these mutations are actually coming from the same set of cells and not different cells, which is shown on the right in the t-SNE plot. And then finally, again, this is a retrospective study, so they went back to their freezer to look at samples. And what they conclude is the clinical status of these samples were actually that there was no morphologic evidence of disease as defined by the percent blast count in these samples. Versus what we show on the right is that you can actually detect these cancerous cells at very low levels. And so really the power of single cell could potentially be, instead of using these large scale morphological ways to diagnose and monitor treatment, you can pair this with single cell technology to get a higher sensitivity and a better look at whether those cancer mutations or cancer cells are still present or not. Okay. So those were our first two publications, and now it's really exciting to kind of see where single cell has launched into. And two really big seminal studies that are coming out, one from Dr. Takahashi's lab at MD Anderson and one from Ross Levine's lab at Memorial Sloan Kettering, has really started doing large scale studies to look at their AML patient samples and do an atlas and catalog them on a single cell level. And so in this study, Dr. Takahashi looked at 77 patients with AML, and he sequenced over 500,000 cells. And I love looking at these plots because it really shows an overview of the whole study in one snapshot. So each column represents a cell, and we can see every single cell from these 77 patients and denote what mutations are coming from each cell. And so what you can see are the different mutations that come from each patient, from each cell, and also whether they co-occur. On the left, there's roughly about 7,500 cells per sample. So again, showing how high throughput the platform can be. And then on the bottom left, there was 100% concordance with bulk sequencing. And again, down to that 0.1% sensitivity, which was confirmed in this case by droplet digital PCR. And so you can take all of this information and reconstruct phylogenetic trees from each patient. On the top are 4 patients, and then on the bottom are 4 patients. And what they show is that the way that these clones evolve and acquire different mutations can be different. Sometimes it's very straightforward, as in linear evolution, where they acquire mutations one by one by one. And then in other cases, there's a more evolved complex branching in which some of the clones branch off and acquire different mutations, while a different branch occurs with different mutations. And what they show is that on the right, when you have branching evolution, you usually have a higher number of clones than when you have linear evolution. And so again, because we now know which mutations are coming from each cell, it's much easier to reconstruct these phylogenetic trees as opposed to having bulk sequencing, where you just know I have 50% VAF of KRAS and 20% VAF of maybe IDH1, and you have to try to infer a lot more in order to figure out how the mutations were acquired based on just the bulk percentage of the VAF. And then you can take this information about the phylogeny of the clones, and you can see how this changes during treatment. So on the top are 2 patient samples that had clinical remission followed by relapse. And so on the left, we can see that this FLT3 clone shown in red was really small during remission. All the clones subsided. And then during relapse, it's this red FLT3 clone that expands. And so again, if you have the power to see or measure this clone early on at baseline, you could potentially treat it much early on in the therapeutic journey. Versus the patient on the right, the FLT3 clone expands, however, it was undetected at baseline. So here are just 2 examples showing how sometimes the clones are there at the beginning of treatment, and when you put patients on therapies, it leads to the diminishment of some clones, but the rise of other clones. Versus on the right, sometimes the clones arise after treatment. And then finally on the bottom are 2 examples of patients that have treatment refractory, meaning that they never responded to treatment. And so again, they were put on FLT3 inhibitors, and you can see on the left, the blue and the purple clone that contain FLT3 go down, but they don't attack the other clones that don't have FLT3 mutations that are still there and causing disease. And the same is true on the right. So they're on a FLT3 inhibitor therapy, the FLT3 clone goes down, but this just leads to a rise in other clones with other mutations. And you can really see this nicely with these fish plots using single cell technology. But what does this all mean? It's nice to create all these really pretty graphs, but I think what's shown really beautifully in the Takahashi paper is that it actually has implications for survival. So what they show is that patient samples that had higher diversity of clones and number of clones had a poorer probability of survival than those that had less diversity in their clones. And next, in a similar study, Dr. Ross Levine also is starting to go back to his samples and do an atlas, a single cell atlas, to look at the DNA mutations that occur in AML. And so his question is, he is very interested in the order of acquisition that these mutations are acquired. So again, this beautiful plot looking at 123 patients, he profiled over 700,000 single cells, and you get this beautiful idea of what the mutations are, which ones are co-occurring. And the right, just like Dr. Takahashi found, he found that as the disease severity increased from left to right, you typically find more mutations in that sample and a larger number of clones. And so he also reconstructed and did phylogenetic trees. And I found what's interesting in this paper is typically you don't get zygosity information when you look at bulk sequencing. But because you get zygosity information with single cell, they had to actually recreate their own bioinformatic tool in order to create these phylogenetic trees that took into account zygosity. And what they show for 3 different patients, again, they see this very linear branching versus more complex branching. And they're able to reconstruct which genes the mutations are occurring in first. And what they found was that the mutations often occur in epigenetic regulators, such as DMNT3A, IDH1, and IDH2 first, before they occur in signaling genes, such as NRAS and FLT3. And furthermore, they found that NRAS and FLT3 are typically mutually exclusive and don't occur from the same cells. So really powerful information. They then layered on the DNA plus protein capability of the Tapestri platform to see that different genotypes have different expressions when it comes to protein surface markers. So in this example, we have 2 clones based on genotype, a DMT3A FLT3 and a DMT3A NRAS. And it's a DMT3A NRAS clone, which is associated with higher levels of CD11B, which is a marker of leukocytes, which is not expressed for the FLT3 mutation. So we really are excited about the potential to pair genotype plus phenotype together and get even greater understanding of what's happening within these clones. So I won't go into detail, but we had the pleasure of having Dr. Ross Levine talk last month during AML Awareness Day. And so I'd encourage you to go to Mission Bio's YouTube channel where you can check out his fabulous talk where he talks about this paper more in depth. And then finally, the next paper we have with DNA plus protein just recently came out in bioRxives with Dr. Adam Abate and Dr. Catherine Smith from UCSF. We're so excited to have the Abate lab next week, next Tuesday, give our virtual seminar series. And so I don't want to steal any of his thunder, but please join us next week where he will talk about this beautiful study in which they profiled AML patients to look at DNA plus protein. So with that, I just wanted to conclude, give you a taste of what is in store with the Mission Bio platform. Very high sensitivity down to 0.1%. The power of single cell gets you the zygosity and co-occurrence of those mutations. Knowing that information, you can reconstruct clonal architecture. We can layer in DNA plus protein and really start to understand what's happening in therapeutic resistance from samples that are coming from clinical trials. We have many more papers from Mission Bio that have come out of clinical trials. And so I'm excited to do a roundup part 2 in which we'll look more in depth of what the clonal architecture is doing when it's under different treatment strategies. With that, I'll thank you for your time and open it up to any questions.
Thanks, Kelly. That was a wonderful review of the Mission Bio publications. So a couple of questions that have come in. I think it's really great data that's coming from these single cell platforms, but often what's asked is orthogonal methods to validate some of this data. Can you speak to what's been shown in some of these papers here?
Absolutely. You know, we had the same questions, our customers had the same question. And so when we were developing our DNA plus protein multi-omics platform, we paired with Dr. Sar Gill from UPenn. And so what was really great working with their group is that they had bulk sequencing data. And so we typically use the bulk sequencing data to compare our results for the single nucleotide variants. And so there was concordance there between what they saw in bulk versus what they see with aggregated single cell. So that's a very typical experiment that people first do when they start doing single cell experiments. The second thing is we looked at copy number variation and we were able to confirm and see copy number variants that are in public databases for that particular cell line. And then the final thing is when we look at the third analyte, which is protein expression, oftentimes these samples have FACS data. And so we can look at the data from FACS to see what proportions of the cells are high or low in different protein markers and see how that correlates with the protein expression data that you resolve with single cell.
Great. Another question here, with analysis, obviously this can always be challenging. What type of tools does Mission Bio provide, you know, either for just some of the variant calling and or, you know, some of these beautiful fish plots that we saw in some of these publications?
We have an end-to-end workflow, and you can see that in some of our earlier virtual seminars in which we go more in depth into the actual workflow and the software solutions. There's primary, secondary, and tertiary analysis. So the primary analysis are the FASTQ files that you get from the Illumina sequencer. And then our secondary analysis is Tapestri Pipeline, in which you upload those FASTQ files and then we'll align them to the genome and we'll deconvolute all the barcodes to get you the single cell information. And then finally, tertiary analysis is when we want to see like, okay, what's the cool, interesting biology that's happening in our data? And so for that, we have Tapestri Insights software, and that allows you to really look at all of the different mutations, the VAFs for the mutations at a single cell level, recreate the clonality from those mutations, and then generate fish plots and phylogenetic trees, all within our Tapestri Insights software.
Great. And maybe one final question here. It looks like many of these publications sort of review heme malignancies, but is the platform capable of working with solid tissue also?
Yes, so we have there's a paper in Cancer Discovery, which we'll profile in the next publication roundup part 2. In which they looked at colon cancer and they looked for mutations and clonality within those samples. We really think that the big applications are heme applications, solid tumor applications, and then CRISPR, genome editing, mouse models. But really, you know, the platform is open, all cells have DNA, and so we're excited to see what our researchers come up with.
Great. Well, thank you again, Kelly. This was a fantastic review of our publications. We look forward to part 2 of the publication roundup in June. And thank you to everyone for attending.
Thank you.